Welcome, class. Before we get into learning how drugs affect the autonomic nervous system, let's take a quick review of the nervous system overall. Everyone has successfully taken A&P, so I know you all know this, but it was a while ago. We know that the nervous system is the control center of the body and that it's divided into several divisions and subdivisions. There's the central nervous system that controls a lot of processes in the nervous system, and the peripheral nervous system, which connects the central nervous system to the limbs and the organs. The central nervous system consists of the brain being the central control and the spinal cord, which sends sensory signals to the brain and motor signals out to the rest of the body. The peripheral nervous system consists of two parts also, the somatic nervous system and the autonomic nervous system. The somatic nervous system controls all the voluntary activities of skeletal muscles. The autonomic nervous system controls the involuntary visceral functions that happen within the body, such as breathing and heart rate, digestion, and sweating. All those are done without conscious effort by the autonomic system. The autonomic system is then further divided into the sympathetic and the parasympathetic nervous system. The sympathetic nervous system controls the fight or flight response. Its main function is preservation, staying alive, or getting out of harm's way. It takes control when the body's under stress or threat. It opens up the airways and increases the heart rate in order to pump more oxygen to the skeletal muscles. It dilates the pupils to increase visual perception. All senses are on high alert. Now, simultaneously, it will also shut down unnecessary functions such as salivation and digestion, because those functions aren't necessary when fighting or running away. This explains why under stress a person will get a dry mouth. Also, part of this response relaxes the smooth muscles of the bladder, which is why the old saying of "I was so scared I peed my pants" is actually true. Now, the parasympathetic nervous system is the opposite of the sympathetic. It's referred to as the rest and digest system. Its main focus is to maintain homeostasis. It's in control when the body is not in panic mode, focusing on digesting food, making repairs, strengthening immunities, and such. That's why you're always told when you're sick to rest, because the body's able to be more efficient and heal faster when it's relaxed. The other thing I want to review is the synaptic junction of nerve cells. This, along with understanding the autonomic nervous system, is foundational to understanding just how drugs work in the body. So let's do a quick review of that as well. The basic structure of a neuron consists of a cell body that houses the nucleus and dendrites that extend outward. The largest extension is called an axon, which reaches out to either another neuron or a target tissue such as an organ or muscle. The axon is covered in myelin sheaths that significantly increases the speed of the action potential down the axon. The end of the axon is called the presynaptic terminal. The autonomic nervous system is a two-neuron chain, as you see here. The first neuron extends from the central nervous system and is called the presynaptic or preganglionic neuron. It then transfers its action potential by way of neurotransmitters to the second neuron, called a postsynaptic or postganglionic neuron. Where the two neurons meet is called a synapse. Some synapses are located within the central nervous system, but if they're located outside the central nervous system, the synaptic group is called a ganglion. Now, these neurons don't directly touch each other. Instead, the preganglionic neuron comes very close to the postganglionic neuron, creating a space called the synaptic cleft. In this cleft is where the two neurons communicate. But because the two neurons don't actually touch, the nerve impulse sent from the brain can't cross from one neuron to the other. So instead, the electrical impulse is transformed into a chemical signal through the release of a substance called a neurotransmitter. These neurotransmitters are synthesized in the neuron itself and stored in vesicles located in the axon terminal. The vesicles respond to an action potential, they migrate, fuse, and then release their contents into the synaptic cleft, where they attach to receptors, which are usually proteins, located on the dendrites of the postganglionic neurons. They don't enter the postganglionic neuron. They just activate its receptors, which then create an action potential, resulting in a nerve impulse within the postganglionic neuron. The new action potential travels down the postganglionic neuron, where it releases more neurotransmitters, which cross the synapse and bind to receptors on the target organ. These receptors are located on and in smooth muscles, cardiac muscles, and glands. The result of this neurotransmitter release will result in either an inhibitory or an excitatory action of the organ, meaning it will either, say, increase heart rate or decrease intestinal movement. Depending on what type of neurotransmitter the postganglionic neuron releases will depend on what the postganglionic neuron does. Once the action potential is created and the cell is at rest, the neurotransmitters are released from the receptor and either re-entered into the synaptic end bulb in a process called reuptake, or degraded by enzymes into inactive metabolites. The whole purpose of neurotransmitters is to carry nerve impulses across synapses.